أهلا بيكم في دروس الأحياء للصف الثالث ثانوي لغات. Today we're going to through the questions of the book about the DNA and nucleic acids. So let's begin. Choose the correct answer. The ratios of nitrogenous bases of nucleic acid of an organism was found to be as follows: adenine 20%, thymine 26%. Here adenine doesn't equal thymine. Guanine 23 and cytosine 31, and both don't equal 2. So this is not a double strand of DNA. This is a single strand of DNA. And they are not RNA because there should be uracil instead of the thymine. Okay, the genetic material RNA is found in rats, wheat, but the virus of AIDS. The codon is a 3 consecutive nucleotides on DNA, mRNA. It's mRNA. The codon is on the mRNA. For if the codon is triplet, the probability of the codon of different amino acids, so it's 4 cubic, 4 to the power of 3, this one. Okay, this is not 43, this is 4 to the power of 3. If it was not a triplet, if it was 2 nucleotides, so it will be 4 square, but it's 3 nucleotides in the codon, so it's 4 to the power of 3. A polypeptide is formed of 21 acids. At least possible number of nucleotides in its RNA would be, it's 21 amino acids, and each amino acid is has a triple codon, so the number of nucleotides will be at least 21 times 3, that's 63. Okay? The following is a part of a DNA strand. This is the 3 end, this is the 5 end. It's adenine, AAA, thymine, TT, cytosine, CC, guanine, adenine, thymine, etc. Write down the sequence of its complementary strands. So adenine base pairs with thymine, so it's TTT. Thymine with adenine, so it's AA. Cytosine with guanine, so it's GG. This is cytosine, this is thymine, this is adenine, thymine, and guanine. Okay, write down the sequence in its mRNA. Instead of thymines, there will be uracil, so adenine UUU. Here thymine with adenine AA, GG, C, U, thymine with adenine AAA, UG, and this is the it will be, by the way, the ends will be the opposite. When you write it down, this won't be the 3 end, this will be start with the 5 end, because the direction is from the 5 to the 3 end. Okay, so this, when you write it down as a complementary strand or on mRNA. Ratio of A plus C and T plus G. If A equals T and cytosine equals G, so A plus C equals T plus G. So this by this will be 1. The ratio will be 1. Gene is formed. Gene X is formed of 150 pairs of nucleotides. What's the number of the amino acids that constitute the resulting protein? If each amino acid is 3 nucleotides, so 150 divided by 3, so it's 50 amino acids. Analyzing the genetic material, virus gave the following results concerning the ratio of its nitrogenous bases. These are the ratios. Uracil equals adenine and cytosine equals guanine. What's the kind of nucleic acid and why? This is RNA because uracil in it. In bacteria, the two processes of transcription and translation occur at the same time. That's true. This is because there's no nuclear membrane surrounding the genetic material. That's true too. So both statements are true, and there is a relation between both. Which of the following statements is not true? The two subunits of ribosomes do not combine except during the translation of mRNA. This is true. The translation of mRNA occurs through one ribosome only. No, this is not true. Through several to over 100 ribosomes attached to it. The mitochondria and ribosomes have DNA. It's the mitochondria and chloroplasts have DNA. So it's not true. The number of tRNA equals the number of 20 amino acids. The number of tRNA equals the number of codons of mRNA. So this is not true. The gene is the protein, no, that decides the appearance of the genetic trait. The gene is special sequence of nucleotides of DNA, okay, that specifies the trait. Give reasons for the following. The position of one strand of DNA is opposite the other to form the most stable combination of hydrogen bonds between the two DNA strands. The ligases enzymes play an important role in the genetic consistency of inheritance of living organisms because they eliminate the changes of DNA with efficiency depending on the DNA molecules that carries the two copies of genetic information. So it repairs DNA, or repairs the changes of DNA. The genome of salamander equals 30 times the human genome, but less smaller number of traits. Because most genome of the salamander is non-coding DNA. Remember the repetitive DNA, it's non-coding DNA, and no ability to form proteins or RNA. The ability of bacteria to degrade the viral DNA, because bacteria produce certain enzymes, attack specific nucleotide sequences in the viral DNA. The presence of the reverse transcriptase in viruses whose genome is RNA to convert own RNA genome or virus of the virus into DNA that can be joined to DNA genome of the host cells. The genetic code is an evidence of evolution because the same codons are translated into the same amino acids in all living organisms on Earth. Okay, the viruses have quick mutations because the genetic material of these viruses consists of single RNA strand, so if damage of RNA strand occurs, it cannot be repaired. Thousands of ribosomes are built in 1 hour because DNA contains about 600 copies of rRNA genes for transcription into rRNA. The polyadenine tail of mRNA is not translated into amino acids because it's there to prevent it from breakdown by enzymes. Proteins are different despite the resemblance of their building units because they differ due to different numbers or kind of the arrangement of amino acids in each polymer, and they're different in the number of the polymers that form proteins, and different in the weak hydrogen bonds that give the protein its shape. Okay, what's meant by each of the following? Plasmid, it's present in some prokaryotic cells. That has a circular DNA molecules, much smaller than main DNA of the cell, is replicated at the same time during replication of the main DNA of the cell and used in molecular technology. Polyribosomes, remember the mRNA attached to many ribosomes. This is called a polyribosome or polysome. A release factor, a special protein, it terminates the protein synthesis by binding to the stop codon. A human genome is the total of all genes in 23 pairs of chromosomes of the human cell. The genetic code sequence of nucleotides on DNA, in particular part of the DNA. The anticodon is the site of tRNA, consists of 3 bases that pair with a suitable mRNA codon at mRNA ribosomes complex. Initiation codon, this is the first codon of the mRNA, consists of AUG nitrogen bases, positions correctly for initiation of protein synthesis, and translates into methionine amino acids. Stop codon is the last codon of mRNA and binds with the releasing factor to terminate protein synthesis. Okay, choose from column B with suit statements in column A. 1. Deoxyribonucleic enzyme, it degrades DNA completely, so 1C. 2. DNA helicase, it separates DNA strands, so it's B. 3. DNA polymerase adds new nucleotides in the direction of 5, this is E. 4. Reverse transcriptase transcribes DNA from RNA, so it's G. 5. Ligase enzyme, it repairs defects in DNA, so it's A. 6. Restriction endonucleases أو هرمونات القطع يعني, it breaks DNA in certain places, so it's D. RNA polymerase, it transcribes mRNA from DNA, so it's F. We'll continue questions next lecture, so I hope you got the maximum benefits from this lecture, and thank you very much for watching.